let's start the session. So I'm Tony Gabaldon from the BSc, and I'm gonna chair this session on genomes. We have three excellent speakers, one proceeding paper and two highlights, and the three of them relate to different aspects of cancer. So we will start with the first speaker, Leonardo Peregrina, who's going to talk about discovering significant evolutionary trajectories in cancer phylogenies. So whenever you want.
Okay. This doesn't work. Yeah. Okay, now it works. Not really. Okay, kind of. Okay, thank you very much. I'm Leonardo Pellegrina, a postdoc from the University of Padova. Very happy to be here and present our work discovering significant evolutionary trajectories in cancer phylogenies, a joint work with Fabio Vandin. Okay, so as we maybe all know, cancer is a disease that is driven by the accumulation of genomic alterations that from an healthy cell give us a selective advantage to the tumor cell, driving the tumor growth into a collection of population of cells, also called clones, that are hit by different sets of genomic alterations. And in fact, one of the main challenges of treating cancer comes from the extreme variability of the genomes of the tumor cells, also known as intratumor heterogeneity. And in fact, it is a crucial challenge to both understand the clonal composition of tumors, but also how tumors evolve. In particular because it is known that the order in which alterations arise has an impact on tumor evolution and on clinical effects of the disease. And this is the problem that we want to tackle in this work. So we want to identify frequent or common trajectories of alterations from cancer patients. Okay, so what we can do is to consider a cohort of cancer patients, and with a combination of sequencing data and methods for phylogenetic inference, it is possible today to reconstruct the evolution of each tumor, obtaining a tumor tree or a phylogeny in which that are essentially trees in which every node is a clone, and that contains all the alterations that are these colored shapes that you can see on the slides that are unique to that clone. This means that if we consider a cell that belongs to a given clone, like the one in purple that we have on the slide, that belongs to the gray clone of the first tree, that cell carries the alterations of that clone, but also all the ones that are on the ancestor nodes of the tree, as you can see on the slide. But still, even if we are able to perfectly reconstruct the history of every tumor, it is still a critical challenge to identify conserved progression patterns of alterations that are important for the disease. And for this particular problem, there have been some recent work that either try to partition trees into clusters according to their similarity, or to only find some simple patterns, such as pairs of alterations with a given order, or linear paths of alterations. However, these methods cannot really discover complex patterns that are typically important for cancer. And furthermore, we observe that the statistical tests used by these methods can be confounded by the co-occurrence of the alterations within the same patient, rather than properly assessing the significance of the order that we actually care when studying tumor evolution. Great, so for these reasons, we developed our algorithm that we call MASTRO for discovering conserved and significant progression patterns of alterations. our algorithm can identify trajectories with complex topologies, as the ones that you can see on the bottom right of the slide, that explicitly encode different types of interactions between alterations, such as the co-occurrence in the same clone, or the exclusivity, so alterations that are on different clones, that are the ones that are on different branches of the trajectories. Also, we developed new statistical tests to properly assess the significance of such trajectories. And also, we used some efficient and powerful procedures to correct for multiple hypothesis testing in order to control for false discoveries, so to provide guarantees on the output, but also to not overcorrect for multiple hypothesis testing by taking into account the correlation between trajectories. Okay. Okay, great. Now I would like to go a little bit in more details on the computational problem that we define and solve. So first, let's start with some definitions. So the alterations that we consider can be any type of somatic alterations of the genome, such as SNVs or copy number aberrations. And typically, we do some analysis by working on alterations at the gene level, but also other types of analysis can be done. And we define a trajectory as a tumor tree, and we say that a trajectory is observed in a patient's tumor tree if all the pairwise orderings between the alterations are the same, both on the trajectory and in the tumor trees. Okay? As an example, in the bottom, I show you a trajectory composed by 3 alterations and 4 nodes. This trajectory is observed in all the tumor trees that we have on the top. This is because we always see first the red alteration, then we see the blue and green alterations on different branches of the tree, as you can see on the colored nodes in the top. And note that the trajectory that we consider do not have to be the alterations do not have to be consecutive in the tumor trees. So we can skip some alterations that maybe are not important for the progression. And this is another example of a different trajectory that we only observe in the first and the last 3. Okay? So these are the trajectories that we care about, and actually we look at a specific type that are the maximal trajectories, that are the ones that contains the largest set of alterations that we observe in the same set of patients. So in a sense, they are the most complete trajectories that we observe in the same set of patients. So the computational problem that we define is the frequent maximal trajectories problem, which we are given in input a multi-set of n tumor trees that we can infer from different types of sequencing data with some previously available method, and a frequency threshold sigma. What we want to compute are all maximal trajectories that we see in at least sigma tumor trees. So in at least sigma patients. Okay? And this is an example of the solution for this problem with sigma equal to 2 for the trees that we have in input at the top. And what we prove in the paper is that this problem is NP-hard. So unfortunately, we are not able to solve it efficiently and exactly for every instance. So what we do is to develop an algorithm that solves this problem exactly, but quickly in practice, okay? That we call Mastro. And the main idea of Mastro is to expand every tumor tree into expanded tumor graphs, in which what we do is to add additional edges to the tree, in which we connect every node of the tree to all the other nodes that it can reach going down into the tree, as we can see on the example on the top right. That is essentially analogous to the computation of the transitive closure of DAGs. And now we have this equivalence between a frequent trajectory and a frequent set of edges of the expanded tumor graphs. And so what we do in order to find frequent trajectory is to enumerate frequent item sets that is a very well-known problem studied in data mining that we can solve quickly in practice. Okay, this is for finding the frequent trajectories, but really what we also want to assess is whether a trajectory is significant in the sense that it is more frequent than what we may expect if the order of the alteration is random on the patients. And so to assess this, we developed new conditional statistical tests in which we condition on the set of alterations of each patient in order to account for different cancer subtypes. And now the null distribution or null hypothesis that we assume is that alterations arise with no order or with random order. This means that if we have, like in the bottom, our tree, that is the original tree that we observe in the data, we assume that it comes from a distribution in which we have the same topology, but let's say a permutation of the alterations on the nodes. Okay. And then to correct for false discoveries, we use some resampling procedures to bound either the family-wise error rate or the false discovery rate by taking into account the correlation of different trajectories. Okay. Now I would like to present some of the results that we obtained with Mastro when applied to cancer data. In particular, we applied it to a cohort of 123 AML patients. These are data from a recent study from single-cell panel sequencing, in which Mastro finds 138 maximal trajectories in at least 2 patients, and can do so in a few seconds, so it is very fast in practice. And what I'm going to show you are the top 40 most significant trajectories that we identify with a small FDR, that are this one on the slides. Of course, I won't go into much detail, but what we can bring home from these slides is that Mastro is able to identify trajectories with complex topologies. So actually, there are some frequent trajectories of these kind of forms that are not simply linear paths, but also with multiple branches and so on. Instead, I would like to see a summary of this trajectory that we can obtain by combining the individual trajectories into 4 major patterns that are shown here, in which every colored box is a single trajectory. And you can see that they overlap into these major progression patterns that are shown from A to D. And interestingly, we looked at some previous works and clinical studies, and we found out that the first 3 patterns are actually known progression patterns to be important for AML, which we have a first mutation in an epigenetic factor, followed by a mutation in NPM1, and later alterations that are clonally exclusive in signaling genes, such as FLT3 and RAS. But in addition, we also found a new pattern that was not previously reported, in which the first event is a mutation in the NPM1 gene that is not preceded by an alteration of an epigenetic factor. And furthermore, these trajectories are observed on different subsets of the patient. So this means that maybe this new trajectory is a different type of modality of evolution. Okay, so to summarize, I present you Mastro. That is a fast and effective method to identify conserved trajectories of alterations from large tumor cohorts, and that is equipped with powerful and sensible statistical tests to assess the significance of the order of alterations, and also on effective and powerful resampling procedures to control false discoveries, so to have guarantees in the output. In the paper, we also show some simulations in which we prove that we show that Mastro is robust in terms of false discoveries with high statistical power. In particular, we show that it can also be applied to smaller cohorts. And on cancer data, we show that it can discover known and new progression patterns. And to conclude, I would like to acknowledge my co-author, Fabio. I would like to thank ECCB for sponsoring my travel here. And also, I would like to say that you can find the code and paper online, and this is the QR code and link that you can use. And I'm happy to take questions.
Thank you. So we have time for questions.
Sorry, just that I don't understand quite well how you get the initial trajectory. So which kind of data you use? You kind of genotype the tumor in several times to know what's the original. Yeah.
So you can use different types of sequencing data, such as single cell data, also bulk sequencing, and of course, if you have like multiple samples, so like a multi-region study that helps in reconstructing the trees. Our method directly starts from the trees. So we assume that you're able to somehow reconstruct the history. Once you have it, you can apply our method to find this frequent trajectory. So it is somewhat independent of the source of sequencing data.
Okay.
In a way.
So
Yeah.
thanks for the talk. I have two questions. The first one is when you do the frequent item set mining over the
Yeah.
edges, how do you make sure that the result is a tree so that it's connected? And the second question is, did I get it right that your test depends on the fact that all patients have the same mutations, which is not very realistic? So you had it in parentheses?
So for the first question, actually, that's a very good point. We have to check that the item set is actually a complete graph in a way. So that is a tree with all the orderings. And so that is something that we have to do while we enumerate the frequent item set. And probably there is a smarter way to enumerate this trajectory, and but that is what we manage. Can you please say the second part again?
In your test, you said it depends on the fact that all patients have the same mutations, not in the same order, but that the set of mutations is
Yes.
always the same, which is not the case.
No. I mean that we fix the set of every patient, so we don't shuffle alterations across patients, but we only shuffle the orderings. This is to avoid that the co-occurrence of alterations make an effect on the orderings. So we don't shuffle alterations on different patients, but only the order. But that's a very good point. Thanks.
Okay, we have time for a last question.
I just wanted to know
Yeah.
if maybe I missed it. What's the minimum and maximum number of items in a trajectory? So how do you define, like, you accept a minimum number of 2 mutations to define a trajectory, up to 5, something like this?
Yes, so now we only consider trajectories with at least 2 alterations. So that I think it is a minimum of 3 edges, I think. Because otherwise, if you only have one single alteration, there is no ordering, and it's not really. But we can also find those kind of trajectories. But we start from at least pairs of alterations with some order. Yeah.
Okay, thank you. We need to move on. We thank the speaker again.
Thank you.
And now we have a highlight talk, and the speaker is Carlos Casimiro, and he's going to talk about towards metagenomics, machine learning interpretable model for understanding the transition from adenoma to colorectal cancer. Whatever you want.
Okay, good afternoon, everyone. I'm going to speak about our last work, where Carlos Lucera and me are co-finished with equal contribution. And the name is towards metagenomic machine learning interpretable model for understanding the transition from adenoma to colorectal cancer. So the first objective of this work was predict the probabilities of a metagenomic sample, whole genome sequencing sample from the human gut, predict the probability of to have colorectal cancer. So we started from these 7 datasets, Hanningham, Bogdan, Zeller, Feng, Yu, Thomas 0, and Thomas 1. In total, there was 1,042 samples and were downloaded from the European Nucleotide Archive. The metadata was obtained from supplementary material and from the conductor package curated metagenomic data. Well, the quality control and the data processing was done with NGLS profile script, where the read with less 45 bases was removed, less with 24 Fed score. The read that aligned with the human genome was removed as well. And finally, we calculated the functional profile. So to calculate this functional profile, we aligned the read against the integrated gut catalog. This catalog has more or less 9.9 million of genes pre-annotated with a GENOMES mapper. So from this annotation, we calculate two functional profiles, one based on GENOMES ortholog and other one based on GENOMES ortholog. And the count for each feature were divided by the feature size. No? So, in summary, every sample is described as a collection of functional modules contributed by the different bacterial species present in it. Moreover, we perform a taxonomy profile with the application centrifuge. So we downloaded the reference genome from archaea, bacteria, virus, human, and mouse, and we aligned the read against this reference genome. So the proportion of the taxon were normalized by the genomic length. In summary, every sample is described by the proportion of each taxon present in it. So we use these 3 types of functional profile as an input for our machine learning pipeline. Then we applied a logarithmic transformation, feature selection by false discovery rate. We discretize this selected feature, and then we use an expandable Boolean matching. This type of matching is a black box. And a new type of general additive model. So in this model, every feature has a sample function, and then all the sample function from all the features are aggregated in the model. Moreover, have a fair attribution. That means that where 2 features are highly correlated, the weight for these features are distributed more or less equally and have additive explanation. So we check the predictive performance for each taxonomy profile, and we do a 20-time tenfold cross-validation. So in the diagonal of the matrix, we can see the intra-project performance, and in the rest of the matrix, we can see the out-of-project performance. We see that we have a good performance for the intra-project, except for Hanningham, but this project was frozen during 25 years before it was sequenced and have the lower dip for his sample. For the out-of-project performance, it's good. Quite well, but we have some projects in between different projects, and we perform as well. We leave one project out. So with training, we leave one project out. We training the model with the rest, So we can see how the performance is quite higher. And moreover, to compare with other papers in the literature, we made out of training selection, leave one project out. So we select the feature and then we apply our model. So we have a good performance, but if we have to extract biological information from the model, we need as well a stability in the feature selection and stability in the rank of this feature. So we check this type of stability with a cross-validation test and with a random stability subsampling test. No? The cross-validation test is a 10-fold cross-validation test, and here the sample were split 50-50 between training and validation set. No? Lower 0.4 is quite random stability, quite random selection. Between 0.4 and 0.7, the selection is good enough, stability is good enough, and over 0.7, the stability is very good. So we can see how the stability in the feature selection is good for all the project except for Hanningham, and the pattern in the rank stability is similar. No? Well, these are some results. This is the top taxonomic feature, and we can see how the first four correspond to two species, the Parvimonas spicra and Fusobacterium nucleatum, and these species are widely used as biomarkers. No? And moreover, Fusobacterium nucleatum is described that it could promote the chemioresistance by modulating the apoptosis of cancer cells. Moreover, we have other genera highly described in other papers related with colorectal cancer, like Porphyromonas, Gemella, Clostridium, and Streptococcus. These are the top features for Kescao taxonomic profile, and here we can highlight 4 bacterial modules. The first one is related with glutamate. It describes that the cancer cells have the glutamate biosynthesis deregulated, so this could promote the presence of this bacterial module in the gut. The second one is related with the excess iron condition, that is as well related with the proliferation of colorectal cancer. The third one is related with the polycated biosynthesis, that is related as well with colibactin, that recently is described that could promote colorectal cancer. And the last one is related with lipopolysaccharide, that with the gram-negative polysaccharide that is present in the third wall of this type of metabolite could promote the colorectal cancer. Moreover, we extracted all the metabolites from Kescao ortholog present in the top feature, and we summarize the score from this Kescao module for every metabolite. And we can get this table where we can see how all these metabolites are described as alterated in a systematic review from Tian et al. Well, moreover, we can see, we wanted to see if the model can learn something about unseen but related condition. We use the adenoma condition for this test, and we use the 50% of healthy sample and the 50% of colorectal cancer sample to training the model. And then we calculated the probability to have colorectal cancer in the rest of the sample, including the unseen healthy and the unseen colorectal cancer sample. And we repeat it 100 times, comparing the distribution. And what we obtain is this radar plot, where we can see how the ECCB profile can separate the distribution from healthy and tumor all the times, adenoma and tumor as well, healthy and adenoma, and small adenoma and adenoma. If we compare this with the taxonomic profile, we can see how it can distinguish or separate the distribution from healthy and tumor, adenoma and tumor, but from healthy and adenoma, these fail to separate the distribution. So this can tell us that the changes in the gut microbiota started with changes in the function, and then with the cancer advance in the patient, promote the changes in the taxonomic level. So these are the top adenoma features, but sadly we can't find any correlation with the adenoma, maybe because there are few studies about or that related with metagenomic sample and adenoma. So the conclusion, the machine learning approach proposed here has demonstrated a more consistent performance in comparison to other approaches previously proposed when dealing with different colorectal cancer-based problems, while providing straightforward interpretation. It demonstrated a better resolution, not only with respect to the separation between healthy and colorectal sample, but it's also able to discriminate samples with adenoma. The model has a biological interpretation that provides important clues to better understand the mechanistic implication of the gut microbiota in colorectal cancer, as well as in the previous stage of adenoma. Thank you for your attention.
Thank you. We have time for questions.
Hi. Thank you for your talk. So as far as I understood, you had several data sets, right, with healthy and sick patients, and then you trained your model on, say, some of them, and you predicted on another one using 3 types of data, right, CAG, AgNode, and taxonomy. Yeah. So I wonder whether you did any emergent approach, wisdom of the crowd approach, when you would take prediction from 2 different data types and then have like assembled prediction, the vote-based prediction.
We did some try in other work, but in this work we didn't try to merge the different profile. So, but in other data sets, in a CAMDA challenge, we tried to predict the location of different cities based on different profile, and if we merge this profile, the result of the model result for this profile, we increase the performance of the prediction.
Yeah, because I just invite you to visit my poster. It's about that if you merge, you get a better resolution and better results. And if you don't mind, I have a second question. So you used IGC gene catalog. I wonder, why did you choose IGC in comparison to UHGP, for example, which is almost twice bigger and better annotated, in my personal opinion.
Yes,
but it's twice bigger. It's not arbitrary, it's for sure.
Yeah, of course. Sure, if we use a better annotated database, the result could be better. We obtained the best result with the GENOMES database that is bigger or have more features than the CAG one.
Yeah, UHGP also has an AgNode annotation. UHGP catalog also has this annotation, by the way.
Okay.
Just, Well, thank you.
Okay, we have time for more questions. Are there more questions? So I have one. So when you use the taxonomy, I see you, because you have metagenomic data, you get really to the strain level. So I see in your table, for instance, you had several nucleatum species, different strains, so which had like high scores. So do you think this is because, anytime you have one nucleatum species, whatever strain it is a feature that is important? And do you think you could gain maybe something by reducing the features by collapsing maybe strains into species? What do you think about that?
Yes, if we collapse the feature into species, maybe the results are similar, because we did it. We create a plot where we can collapse the feature, and we collapse as well the score, as well the principal feature were Parviomonas migra and Fusobacterium nucleatum.
Okay. Any other question? If not, we thank the speaker again. Thank you. And the next speaker is Solid Park. It's also a highlight talk. And she's going to talk about higher order genetic interactions, switch cancer genes from 2-hit to 1-hit drivers. Whenever you want.
Hello. Hello, everybody. I'm Solip. I'm very happy to present our research project, especially this project is collaboration project among 3 different research centers in Spain. I'm from CNIO in Madrid, and from IRB in Barcelona, and then from CRG in Barcelona. So I think it's very nice to present this work in Sitges. So as you know, it's quite incredible development of sequencing technology. So it's not so difficult to produce huge number of cancer patient sequencing data, like 100,000, 10,000, something like this. So based on this large-scale analysis, we found a very interesting, exceptional example beyond the classical cancer gene model. For example, we can distinguish the cancer gene into 2 groups. The first one is tumor suppressor, and the other one is oncogene. So people usually thought that the tumor suppressor gene, they usually follow the 2-hit model. It means they are biallelic inactivation. And also, we thought that oncogene might follow the 1-hit model. It means a single mutation or copy number change is enough to increase cancer fitness. However, we found lots of interesting exceptional case. For example, 1-hit tumor suppressor, it means haploinsufficient tumor suppressor, or obligatory haploinsufficient. It means 1-hit is okay, however, 2-hit does not give any kind of beneficial effect. Or we also find some interesting exceptional case. In the case of oncogene, it means mutation and copy number gain or amplification can be incorporated, can be co-occurred together. So based on this kind of interesting exceptional case, we have very simple question like all the cancer genes should behave according to the same model. Like for example, tumor suppressor gene should follow always a 2-hit 2-hit biallelic inactivation, or oncogene always follow 1-hit model. So you can imagine BRCA1 has the same mutation at the same position. However, their effect can be different when they are in the breast cancer or they are colon cancer. So we think the different cellular context, for example, tissue, might be the very important factor that can change their model. So to answer this question, we designed very, very simple model. So you can imagine there are 3 possibilities like this. So there are normal cell, and then there's one somatic mutation can accumulate. And the first possibility, they just follow 1-hit model. So it means a single mutation is enough. And the other possibility, like a 2-hit oncogene, means a mutation and the copy number amplification can be co-occurred. And the other possibility of the 2-hit tumor suppressor gene, it means mutation and the copy number deletion can be co-occurred. Okay, this is our simple example to understand the model. So to understand this model, we measure the co-occurrence between the mutation and the copy number change within the same gene. So it's kind of cis interaction. Okay, so we want to quantify they are co-occurred or not using regression model. So for this model, we use the TCGA data. As you know, like it covers more than 10,000 tumors across 30 different cancer types. And we specifically collect more than 200 somatic driver genes. They are highly well-known and high-confidence somatic driver, including like 53, LAS, VRCA1, like this. And also, we are specifically test like their targeting cancer type. For example, BRCA1 and 2 test ovarian breast cancer, 53 test a lot of different cancer types, something like this. So using regression model, we quantify the mutation and the copy number change within the same gene. They are significantly co-occurred or not. Okay? So this is the simple regression model to measure this. So you can imagine like there's two different models because as you know, the copy number change can be classified into copy number gain or amplification or loss or deletion, okay? So we apply two different copy number class like a loss or deletion or gain or amplification. So you can, this is just a toy example. I test 3 different genes across different cancer type. You can see different size of circle. It means they are highly co-occurred or no interaction, something like this, okay? So I would like to show like a diversity of the interaction across the cancer type. So this is our observation when we test our regression model across a lot of somatic driver genes across the cancer type. So first impression that you can find here, you can see there are huge difference between the gene and across the cancer type. So you can see that many tumor suppressor genes shows strong interaction with the mutation and deletion. However, their preference is very different. You can see that some of them shows very, very, very strong co-occurrence between the mutation and deletion. However, some of them shows like 30 or less than 30, something like this. So we can observe the similar pattern in the case of the oncogene. You can see that EGFR, they are always co-occurred with the mutation in the copy number change. However, like NLAS or VLAF, like there are sometimes like 30% of them shows co-occurrence. And one of the most extreme case, like 53 or this kind of gene, they shows like interaction, either mutation, deletion or gain, depending on cancer type. Okay, this is kind of a very extreme case. So to summarize this, we can find very interesting diversity across the cancer type. Some of them consistent to hit or mild, or some of them shows a mixed class. And we also find some somatic driver always follow two-hit model. So this is a kind of systematic view. I would like to introduce like individual case. So this is our overall analysis when I apply regression model across the gene and across the cancer type. So to understand this systematic view better way, we define the 4 different class, the class 1, 2, 3, 4. For example, in the case of the class 1, it means we test a lot of different cancer type. However, they didn't show any kind of significant interaction, either deletion or gain. Okay, so it covers a lot of different tumor suppressor and oncogene. And interestingly, we found that they are highly enriched to the essential gene. And we assume that that's why they try to avoid a strong interaction with the mutation and copy number chain. And the other class 2 and 3, class 2 means 2-hit mutation and loss, and class 3, 2-hit mutation and gain. And as you can expect, like a lot of tumor suppressors are belong to class 2, and a lot of oncogenes are belong to class 3. And also you can see, as I told you before, that their circle size is very diverse, like very strong and small, like also here as well. So you can see clearly like their interaction, the strength is very, very different depending on the cancer type. Okay. And the last case is one of the most extreme and complex case. You can see that some 53 or these 3 genes, they show interaction both in the loss and the gain model, depending on cancer type. So especially in the case of 53, it is well known that they have a tumor suppressor effect and oncogene effect depending on mutation position or depending on their cellular context. So we expect that this is because we observe this kind of pattern. Okay, so from this kind of analysis, I would like to show that their interaction between mutation and copy number chain shows a lot of diversity. And then the next question, like how they can change or switch this kind of two-way and one-way interaction depending on cancer type. So the next question, we have some hint. So as you can imagine, like all the cancer patients, they usually have more than one different somatic driver mutation. Of course, the number can be different depending on cancer type. For example, the kidney cancer, they known to have 1 or 2 different somatic driver mutation. And the colon cancer is one of the extreme case, like they usually have like more than 10 different somatic driver mutation. So you could imagine that there are lots of possibility they could make another interaction between 2 different somatic driver, okay? And that's why we are very interested high order interaction or 3-way interaction. So our definition of 3-way interaction means that we already observed 2-way interaction, and when we consider another somatic mutation from different gene, depending on the mutation state of different gene, the 2-way interaction can be perturbed. It means they can be more strong or less strong, okay? So this is our definition of 3-way interaction or high-order interaction. So we apply similar regression model, but we added one more genomic alteration feature from another gene B, okay? So we are using 3 different factors like mutation from gene A and the copy number change from gene A and the mutation from gene B, okay? So this is the regression model to detect the 3-way interaction. So this is a very well-known or interesting example to explain the 3-way model. It is very well known that BRAF mutation and the BRAF copy number change, they are highly co-occurred each other in the melanoma cancer, okay? And when we test 3-way interaction model, including N-last mutation, this strong interaction can be disappeared depending on the N-last mutation. Okay, so this could be very representative example to explain what is the 3-way interaction. So you can see the 2-way interaction can be decreased depending on the N-last mutation. So when we systematically analyze the 3-way interaction, we can identify more than 10 3-way interaction partners. And one of the interesting points from the 3-way interaction, we found that a 3-way interaction partner, they are highly located in the same signaling pathway, or they share the signaling pathway or biological function or cellular location, okay? So they could be kind of redundant pathways like this. And we also mapped the 3-way interaction to the cancer representative signaling pathway, and we found that most of them belong to very well-known cancer-related signaling pathway, like last signaling pathway or 53 pathway or something like this. So we expect that this kind of 3-way interaction partner, they could play a very important role in the cancer progression. So I would like to introduce what kind of ongoing thing in my lab based on this observation. So related with the first talk, cancer evolution is quite important to understand cancer progression. So especially you can imagine like the germline variant can be appear at the first time and later somatic mutation, and the loss of somatic mutation also can the metastasis. So I would like to add this kind of time event because I'm talking about high order interaction. So it means that something could be triggered for the high order interaction. So there are 2 projects ongoing in the lab. For example, Manuel is working with the high-order interaction between germline variant and the somatic mutation, and Laia wants to understand the high-order interaction difference between the primary and the metastasis. Okay, for example, in the case of the second project, we are using MSK-MET data because it's really big data. It's highly balanced between the primary and the metastasis. So, for example, we found that there are significant difference from the primary metastasis. So when we apply the similar regression model, like a mutation loss or a mutation gain, we compare the interaction strength between primary and the metastasis. You can see that the sum of cancer types shows significantly different interaction strengths when they are primary or metastasis. So I hope that this kind of analysis will give some hint to understand the difference between the primary and the metastasis, or to understand the cancer evolution. Okay, this is the last slide of my talk, and thanks for all the lab in my lab and also collaborator of this paper, and thanks for listening, and any question will be very welcome. Yeah, thank you.
Okay, thank you. We have plenty of time for questions. I don't see a question there. Yeah,
Okay,
go ahead.
hi. Thanks for the nice talk. I was wondering, it's a very short question. So when you focus on interaction between deletions and mutations, is it only deletions of the whole gene, or you also consider as deletions things like premature subcodons or stuff like so, truncating mutations?
Okay, so when I say deletion, it's only consider copy number change.
Okay.
So like, for example, like when you say a truncation, blah, blah, this could be belong to mutation.
Okay.
Okay, yeah.
But for example, the effect of a deletion could be the same as the one of a truncation in the concept of interaction, right?
Could,
so I don't
yeah.
know whether that would
But
be
in the terms of definition, yeah.
Okay. Thanks.
More questions?
Hey, and thanks for the talk. I have a question concerning like whether you have considered confounding factors like cancer subtypes or like clinical phenotypes that may also affect your inferences of interactions. And second question is that whether your model will be affected by a sample size. So sample size, for example, the statistical significance may be restricted if the sample size is too small. So these are two questions.
Okay. So I will answer the second question first. So you are talking about the sample size. So of course, all the analysis, I guess, we always have some issue or limitation with the sample size, okay? So in the case of TCGA, the sample size is very diverse from less than 100 to 900, okay? So in my analysis, I only include the tissue. The sample size is bigger than 200, okay? So this is the way of try to avoid the issue, like the small sample size issue. Of course, this could be biased when the sample size is enough or high. It could be more better to identify this kind of enough number of sample size. So to answer your question, it could be, but I try to avoid this problem to put some range of the sample size, okay? And about your first question about the subtype or phenotype, something like this. So if I understand correctly, your question, like always the cancer subtype could be, how can I say, like a problematic factor to analyze the co-occurring or mutual exclusive or something like this. Because, for example, I'm measuring the co-occurrence or mutual exclusivity based on regression model. However, this is just because of they are from like a different subtype or something like this. Because my analysis, I did not consider subtype, okay? So why I did not consider subtype, because related with your second question, if I apply subtype, the sample size will be, it decreased a lot. So that's why I did not consider, but this time I did not check But however, before or later, I checked that how much my data can be changed depending on subtype, something like this. But I found that, let's say, 60 or 70 of the interaction that I observed are very consistent. It's not a subtype-specific interaction. However, minor of them could be, due to the each subtype for sure. Yeah.
One more question here.
Yeah, interesting talk, thank you. Kind of related to that question, I just remarked that I'm slightly surprised that you have the statistical power to analyze three-way interactions, given your sample size. Could you comment?
So, okay, so that's why we could not observe many three-way interaction, okay? Actually, we test our input for the three-way interaction model more than 1,500 or something like that. I test 1,500 pair, but FTR 10%, I can say only 30 three-way interaction can be detected. So it means the statistical power is really low. So this is due to the sample size, or like the problem itself, three-way interaction is really difficult to detect. So I don't know. So my plan, like, I'm wondering, like, if I can test like a bigger sample, what can I test more, or if I can increase the resolution, for example, if I use more refined or like a high qualified somatic mutation or something like this. But anyway, right now, I think my data is quite underestimate of the three-way interaction because of the technical issue.
Okay, thank you.
Thank you.
Okay. No more questions, so join me in thanking all the speakers for this session.